Welcome to Biology My Passion. I am Soumya Harikrishna. Let's move on to the classification of fungi and then we will learn one by one in detail. Fungi can be classified into different classes like phycomycetes, ascomycetes, basidiomycetes and deuteromycetes. As I always say, first learn the names in order, then the characters will be easy for you to remember in a sequence because always it's a gradation. So first, what is it? Phycomycetes. Second, ascomycetes. Third, basidiomycetes. 4. Deuteromycetes. The study of fungus is called a mycology. Okay. Now do one thing, pause the video for some time or maybe 1 or 2 minutes and repeat these 4 a few times. Phyco, asco, basidio, deutero. Okay. Deuteromycetes is otherwise called a fungi imperfectly. Something is not perfect there. We will learn. Now, what is the basis of this classification? Always it is good to know what basis we are classifying so that it is easy to remember. So first of all, what is the type of mycelium they are having? Mycelium means what? Tuft of hyphae. And mode of spore formation. Is it exogenously produced or endogenously produced, or which type of spore it is produced? And fruiting bodies. I told different names are given to different fruiting bodies. What are fruiting bodies? Spore bearing structures. But that depends upon the class they belong to. Now let's see one thing, the hyphae, how they are different. So we told hyphae is a thread-like structure, right? So if I draw a hyphae like this has got cytoplasm and it has nucleus also in it. The second hyphae I am drawing with a cell wall, nucleus, cytoplasm. Now any difference you can feel? Yes. The first one, everything is in the same compartment. There is no wall between them. So we call that wall as septum. Whereas we have learned septum in the heart. The wall of the heart separating left and right side is called septum. The same way in nasal septum we have. Okay, septum means it's a cross wall. So here aseptate fungi. Okay, aseptate. So this has septum, so it is septate. This is also septate. This is also septate. Now aseptate it is. So all the nuclei are on the same cytoplasm. So do they have uninucleate structure? No. They have multinucleate structure. So that is called a cenocytic hyphae. Cenocytic means multinucleate. The hyphae have many nuclei. Rest all are uninucleate because they have septum separating. One cell has only one nucleus. Understood? So this is the main difference in hyphae. Only remember this. Only the first one has aseptate and a cenocytic hyphae. Rest all have septate and a uninucleate hyphae. Okay. Now we will have to learn one by one the type of spores and how they reproduce. Phycomycetes are found in aquatic condition, or they are mostly found in damp places or decaying matter because they are acting as decomposers, or they can be parasites on plants. Next, their body, as I mentioned, mycelium is aseptate, that means without septum. As a result, it is cenocytic or multinucleate. Then coming to a mode of reproduction, it is through asexual reproduction or sexual reproduction. Asexual spores are zoospores or aplanospores. These zoospores, what is the difference? Zoospores are motile spores, aplanospores are non-motile. But what is the similarity? They both are endogenously produced, means they are produced within a sporangium. Then next, coming to sexual spores, the gametes can be isogamous, anisogamous or oogamous. Let's see how the reproduction takes place. Actually what happens, suppose this is one hyphae, I am not drawing the cell wall, this is another hyphae. I will draw with the red. This is another hyphae. So they are genetically different, but compatibility is there, mating compatibility should be there. So this is plus, this is minus. Now what happens, we know that it has a multinucleate structure, many nuclei are there in the cytoplasm, cenocytic condition. Now from here, a small outgrowth will come, okay, and the nucleus also will move into that outgrowth. The same thing happens here also, the outgrowth comes on the same side and the nucleus moves into it. Now this is called a progametangia. Your textbook does not given in detail, but those who are preparing for NEET can see these points. Now, after this, a wall will form here, separating this structure from the previous one. Now we call it as gametangia. Means they are separated. Okay, so uninucleate. Now what happens? In the next stage we can see. Next stage we can see these two are fusing. Okay, they both fuse together and they form a structure. Okay, so when they fuse, I told already, first a plasmogamy, fusion of the protoplasm, which results in a dikaryon stage. This dikaryon will fuse to form a structure which is zygote, which is diploid. Okay, now what happens? The zygote will make a thick wall around it. Then we call it as zygospore. Okay, now I am drawing it little down here. Suppose this is the zygospore from both this. This zygospore has a diploid nucleus. Now this will undergo reduction division and this is actually forming a sporangium. This is the sporangium. So this stalk of the sporangium is called a sporangiophore. That is, sporangium bearing structure is called a sporangiophore, and this is sporangium. Spore bearing structure is called a sporangium. Sporangium bearing structure is called a sporangiophore. That's a stalk. Now this will go up and the nucleus will undergo reduction division to form what? Reduction division means what? Meiosis. So that the diploid nucleus will become haploid. But many are there. Now they all will come to the side and accumulate cytoplasm and they are released out. Okay, some may have one nuclei, two nuclei, three nuclei, many nuclei will be there. Now what happens once the conditions are favorable, they develop into the new hyphae. Understood? Yeah, these spores can be, sometimes they can be plus or sometimes they can be minus. So they will produce two types of hyphae, plus hyphae and minus hyphae. When they come together after maturation, again they can undergo reproduction or the sexual reproduction. This is how sexual reproduction is happening. And the important members of this group are, one is the bread mold. What is the name of the bread mold? It is called a rhizopus, which is causing the damage or the making the bread stale. Then after bread mold we have another member of this, mucor. Another filamentous fungus like this is a mucor, or we have another one, albugo. Albugo is a parasitic fungus on mustard plants. On the leaves of mustard you may see white patches. Those are due to infection by a fungus called albugo. So the examples in each class is very important. Mucor, rhizopus and albugo. Rhizopus is the bread mold, albugo is the plant parasite. Okay, so two hyphae come together, plus and minus. First they produce a protuberance called progametangia. One nucleus enters in it and a septum forms separating it from the rest of the hypha. Then it is called a gametangia. The gametangia fuse to form a zygote. Now nucleus also fuse, that is karyogamy happens, resulting in a diploid zygote. Zygote will develop a thick wall to form zygospore. Zygospore will germinate to form a sporangium. The sporangium inside the sporangium the nucleus undergoes meiotic division to form many haploid spores. These haploid spores will be released out, which will germinate into new hypha. Hope you understood all these concepts very well. If so, please like, share and subscribe to my channel, Biology My Passion.